Hello, everyone, and welcome to this new Gen webinar entitled Right First Time, Successfully Progressing Biotherapeutics from Discovery to the Clinic. This webinar was made possible by a sponsorship from Lonza. I'll be your host for today's presentations. I'm Jeff Bogaliscus, the technical editor for Genetic Engineering and Biotechnology News, which has been covering the latest tools and trends in the life sciences industry for 4 decades. And today we bring you a webinar that's going to dive into some strategies that can help successfully drive a biotherapeutic candidate toward the clinic. Throughout the drug development process, it's critical to evaluate potential liabilities as early as possible, for instance, during molecule design. Then, by utilizing lead selection phases, the chances of identifying a successful candidate will increase significantly. This approach puts a special emphasis on identifying the best approaches and tools in order to maximize success when moving the selected candidate through development. Now, here to tell us more about these strategies are our 2 guest presenters for today's event. It's my pleasure to introduce our first speaker, Dr. Yvette Stallwood, who is Head of Applied Protein Services at Lonza, where she and her team are focused on the development and provision of services to support the development of new biotherapeutic proteins and vaccines, with a particular focus on immunogenicity, manufacturability, and protein expression. Today, Yvette will tell us more about strategies to optimize the amino acid sequence of candidate molecules, as well as discuss techniques to maximize product safety and quality. Next, it's my pleasure to introduce Dr. Allison Porter, who is the Head of Expression System Sciences at Lonza, where she and her team provide support and guidance for technical challenges and questions that arise at any stage in the production of biotherapeutics within the organization. Today, Allison is going to tell us more about how the GS piggyback system can help improve protein expression levels, especially for more complex next-generation biologics. But before we begin, as always, I want to remind the audience to stick around for the Q&A session that will occur right after our second talk. Don't be shy. You can ask a question at any time during the presentation, and we're going to try to get to as many of your questions as possible, so keep sending them in. To ask a question, it's really very simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and then hit Submit. Like I said, we're going to try to get through as many of those questions as possible, so keep sending them on in at any time during the presentation. Okay, with all that said, now I'm going to turn the mic over to Yvette to get things started.
Hello, and welcome to this webinar by Lonza on Right First Time, Successfully Progressing Biotherapeutics from Discovery to the Clinic. There will be 2 presenters today. My name is Yvette Stallwood. I'll be starting first, and then I'll be handing over to my colleague, Allison Porter. So I'd like to start by introducing you to a number of different tools which can be used to support the prediction and mitigation of risk of failure in developing therapeutics. A number of these different tools can ideally be performed really to support the late-stage discovery and early-stage development of your therapeutic. So I'd like to start by introducing the concept of developability assessment. Well, to be fair, I'm sure many of you are very familiar with the concept, but really, why is it needed? Well, only around 1 in 1,000 preclinical candidates will successfully reach the commercial market. A number of these failures are due to lack of efficacy and safety, including immunogenicity. Issues linked to stability and aggregation can greatly increase the cost and delay your timelines of development. So there are a number of new technologies which can be utilised to reduce the attrition rates and the cost of development. So I'm going to introduce and talk to you about some of these today. So really, with developability assessment, this allows us to address these issues early and to reduce the risk and maximise the outcome of being able to successfully develop your therapeutic. Ultimately, it's really about knowing as much information about your product as early as possible. So I'll start by introducing the toolbox that we have within the Applied Protein Services team. As you see here, a number of different tools largely grouped into 3 core areas, looking at different aspects of developability. So we're starting with immunogenicity. So we have both in silico and in vitro tools that allow us to predict and understand the risk associated with the immunogenicity of the product. Looking at manufacturability, we have a number of different tools which look at aggregation risk and the risk due to post-translational modifications. Then, of course, protein production. I'll be talking about this, just a gentle introduction. Alison will be talking in much more detail. But ultimately, with all of these, obviously, the more tools we can perform, the better, because it helps you to de-risk your molecule more. But really, the concept of this is that we use these tools during the late stage discovery, early stage development, and it really helps to de-risk your molecule before you're making the commitment to cell line development and obviously moving through development, manufacturing, and of course into clinical trials. So let's start with an introduction to manufacturability assessment. So it's a purely in silico tool that helps us to identify potential high-risk areas in your molecule based on both a combination of sequence and structural analysis. So this is just a snapshot of some of the things we're looking at within the tool. Things such as post-translational modifications, obviously the presence of things such as deamidation sites, glycosylation, oxidation. If they do materialize, they can really impact the stability, efficacy, and safety of your therapeutic. Obviously, protein aggregation can also significantly impact manufacturability. And we look at different areas of protein aggregation dependent on the product type. So with this tool, we start with the sequence, and we're looking at motifs and regions within the protein that indicate, the presence of a deamidation, for example. But we also look at this in the context of the structure. This allows us to understand and interpret the impact of the site if it does occur. Obviously, if it's something which is buried in the core of the antibody, for example, it's highly unlikely to go on and impact the stability and efficacy of the product. Whereas obviously, if it's perhaps in the CDR of the antibody, obviously it can cause a lot more harm to the product and development. So being an in silico tool is relatively quick. We can perform, these sequences, say, analyze 2 to 5 products in 1 to 2 weeks. But it really does add value to your program because it means that potentially at a really early time point, if you had a really nasty molecule in there, you could stop working with it at a really early time point and just focus on the lower risk candidates. An example here where we had an antibody that had an aggregation issue. You can see here, if I get my pointer, on the left-hand side here, we had a wild-type antibody which had around 9% aggregate. This is straight after protein A purification. So a reasonably high level that obviously needed addressing. And you can see here just 1 or 2 amino acid substitutions within the full length of the antibody. It's been enough to substantially reduce the aggregation risk. We've taken it down from around 9% to just a couple of percent, so a much more acceptable aggregation profile. Of course, one of the upsides of being able to mitigate the aggregation problem, we may often see an increase in production. So as we see here, an increase in titer with a number of variants where we perform substitutions to reduce the aggregation. But ultimately, of course, when performing any kind of protein engineering, it's important that the profile of the product isn't impacted, which is exactly the case as happened here, a number of different substitutions, but the binding and affinity was retained. But in essence, examples such as this really demonstrate that it's taken a high risk candidate through to a much lower risk, but obviously the earlier the better this is done. This could be done before you even start expressing and even really thinking about cell line development. So get rid of the problem at the outset, correct the problem within the sequence, and you have a much smoother process through to cell line development and on. So moving on to another aspect of developability that we should all look at, of immunogenicity. So starting with our in silico tool allows us to predict potential T-cell epitopes. I'm aware not everyone will be familiar with immunogenicity, so I'll start with a very quick introduction to it. So basically, immunogenicity is the intrinsic capacity of a product to introduce an immune response in a target population. It's important that this is considered not just for therapeutic proteins, but also for vaccines, immunotherapies, and cell and gene therapy, for example. I guess a couple of key differences. Really, with therapeutic proteins, immunogenicity is certainly unwanted. You do not want your product to go and raise an unwanted immune response once you've delivered it to your patient. Of course, with vaccines or immunotherapies, you want the opposite. You want your product to be as immunogenic as possible. But a number of these tools can be used, for both therapeutics and for vaccines. In essence, they're used exactly the same way. We're just looking for different answers. So a number of different drivers for immunogenicity. We kind of largely base into kind of product and process related factors and clinical factors. So starting with product and process related factors, or the product. The presence of host cell proteins and aggregates can certainly influence the immunogenicity of the product. Formulation excipients, maybe something is being added to the buffer in the formulation to help stabilize the molecule, but potentially this could actually be making it more immunogenic. Looking at the protein, well, what species is the product? Is it human, humanized, maybe a chimeric, for example? The presence of B and T cell epitopes, the regions of protein which can go on and induce the immune response. And then, of course, post-translational modifications. Again, the potential glycosylation issues or deamidation, can influence the immunogenicity of the product. So we will quite often perform these two different in silico tools at the same time, in a really early time point, get rid of the higher risk candidates. Then moving on to clinical factors. Let's not forget the patient. We all have different immune statuses and different HLA types, which will make some of us more or less susceptible to raising an immunogenic reaction to the therapeutic. And then lastly, the treatment. The route of administration can influence how immunogenic a product is, as can the frequency of the dose. So immunogenicity has gained quite a lot more prominence in the last couple of years. Here's a snapshot from the FDA to get a regulatory perspective on this. And the take-home message from this is that the sponsors should provide an immunogenicity risk assessment to support their IND package. We've certainly had clients who've come to us in the last few years who've reached the stage where they're ready to submit, and they've basically been told they need to go and get some additional data to look at the immunogenicity risk of their product. Some of these assays, the in silico and the in vitro, they can be performed at a really early time point. You don't have to wait until you've got your clonal cell line material or from your manufacturing batch. You can do it at a really early time point to support your lead selection and your de-risking strategy. What we absolutely don't want is for products to be ready to go into phase 1 and then discover they've got an immunogenicity issue. So start with an introduction to the in silico tool. So this is based on the EpiBase platform. And really what we're doing with this tool is identifying the presence of potential T cell epitopes within a protein. This tool is applicable to any kind of recombinant protein. So we start with the amino acid sequence of the protein. And the tool works by cutting this into 10-mer overlapping peptides. Each of these peptides is potentially a T cell epitope. So we identify the potential T cell epitopes. We then identify the HLA receptors to which these epitopes are predicted to bind. The binding strength itself will also influence how immunogenic a product is. The tool will also take into account the promiscuity of binding. If we have an epitope which is binding to a larger number of HLA receptors, this potentially increases the immunogenicity risk, as does the frequency of the HLAs in the population. If we have an epitope which is binding with strong binding affinity to an HLA receptor which is really frequent within the population, it's obviously putting a greater number of the population at risk of raising the unwanted immune response. What a lot of our clients are looking for is the overall risk score, as we see here on the right-hand side. In very simplistic terms, the higher the score, the greater the immunogenicity risk, because there are simply a higher number of epitopes present, but also the epitopes which are present, they're at a higher level of risk. But we shouldn't always just go exactly based on the score. Obviously, the type of molecule involved will also influence the immunogenicity risk. But this really gives you a very simplistic view of how it can work. We see here 5 different antibodies: fully human, humanized, chimeric. Of course, a chimeric antibody, as we would expect, has the highest risk score, in essence because it has the greater number of T-cell epitopes which are present. But you can see here at the bottom really is quite a broad distribution of scores, even just within the antibodies. And I think some of you may be surprised to note that even fully human antibodies really can have potential immunogenicity risks. Now, I'm often told that, oh, my antibody is fully human, I don't need to worry about immunogenicity. Well, I'm afraid to say, yes, you do. It really is important, irrespective of what the product is, that we really need to be considering immunogenicity at an early time point. So that leads me nicely into our light path discovery. This is really Lonza's protein expression platform that supports the lead selection and characterization phase. At this stage, everything is non-GMP. We really are supporting the research activities. Within the APS team, we can actually, and to be fair, within Lonza as well, we can perform expression within CHO, E. coli, or Pichia. So really, the expression platform is really to suit any kind of product. Within the mammalian platform, we're going to be using the Lonza GSX-Seed platform, and I know Alison is going to be telling you more on that later, so I'm not really going to go into too much detail. But because of the amounts of material we're generating at this stage, it is only to support research. It's typically using transient transfection and stable pools. We want to get material as quickly as possible. We can then use our Lonza excess technologies to look at perhaps microbial or yeast with Pichia, really dependent on the product type in question. So it's just an overview of the capabilities and experience within my team, the APS team. A lot of experience, as you see there, well over 1,000 different products expressed by transient or stable pools. And we see roughly a 50-50 split based on mAbs and non-antibodies. Certainly the number of non-antibodies is certainly growing and becoming a much higher percentage over the most recent couple of years. But an overview of the capabilities, and this is really just the transient transfection and stable pool activities I'll take you through. You know, quite often with these programs, we're producing material from a number of different variants, maybe between a couple of variants, again, up to perhaps 10 different products, and really generating varying amounts of material from a couple of milligrams to a couple of grams. Typically material is used, maybe to support some in vitro assays and functional efficacy studies, perhaps through to some early in vivo efficacy models. So we are using Lonza's CHO-K1SV GS knockout platform, the GSX-Seed. So these are suspension adapted cells where the GS gene has been knocked out. Even at this stage, everything is animal component free. We have a number of different vectors which are available to us. So we can use a vector for anything from a single gene to a 4-chain multi-gene with 4 different genes or copies of the protein, or sort of copies of a gene, I should say, within the single vector. Each chain or gene is under the control of its own copy of the CMV promoter. This allows us to be much more flexible with the type of product that we can express. Again, I know something Alison is talking on a little later, but certainly puts us in good position to support the more complex bispecifics or perhaps a protein where we need to co-express a cofactor, for example. I think it is worth noting that even though this is work we're doing really to support the late discovery, early development, the vectors that are created here are going to be exactly the same vectors which would go on and be used for a cell line development program. So I already mentioned that at this stage we're only expressing relatively small amounts of material in the grand scheme of things, a couple of mgs up to a couple of grams. So we'll typically use either transient transfection or stable pools. Purification at this stage, relatively simple. We don't need to go and do a full-on purification development program. For something such as the mAb, it probably is only going to be a one-step protein A, but we can obviously do a tailored approach dependent on the type of molecule. And product analysis at this stage, relatively simple. We're really just demonstrating the product express, the size, and perhaps looking at charge, but we can be flexible with what we include here. Obviously, with things such as an antibody, we pretty much know which expression system is needed. It's going to be a mammalian and CHO-based system. But sometimes, for some of the more unique products, perhaps some of the smaller antibody fragments or recombinant proteins, it's not as clear-cut. And this is what we offer here with our rapid host screen. It basically allows us to either work with 2 or indeed all 3 different expression systems in parallel, generating really small amounts of material, but allowing us to really assess which platform is best. Sometimes kind of our clients think they know which one they really want, but they can't quite, kind of rule out working with one of the other expression systems. And this is what this tool allows us to do. So to finish off, before I hand over to Alison, an introduction to some of our in vitro immunogenicity assays. So really the next step beyond from in silico. So with these assays, we're looking at the whole product, so we can take into account any of the perhaps production impurities or the impact of aggregates or post-translational modifications, for example. So within this platform, we have a number of different assays, which, as you see here, really can be used to assess the 3 main aspects of the immune response. So immunotoxicity, the innate, and the adaptive immune response. We certainly do not need to perform all of these wide range of assays for your product. They really kind of fit in at different times of development. I always tend to say, we work with you to pick the assay that really addresses the question that you want addressed and answered at a particular time point. So assays linked to immunotoxicity, as you see there, they can look looking for immediate response due to the direct product mode of action related events or due to target mediated activation. The assays around the innate immune response are looking at short-term response, as you see there, to product or process-related impurities. And then assays associated with the adaptive immune response, really this is going to represent the longer-term response that's induced by the product. And these include the naive CD4 T cell responses that's associated with the generation of the anti-drug antibody response. So I should say that you'll have a number of different platforms that look at both CD4 and CD8, and we also have a number of different B cell assays. Just one example here of some data. Obviously it's important that the data generated in these in vitro assays is representative of clinical data, and this is what I'd like to show here. So you can see here we've got 4 different therapeutic products which we've tested in our assays, and we range from a very low level of response, an immunogenic response, less than around 5% of our donors responding, up to around 30% or so, so quite a broad range. But what we are seeing is that the levels of donors responding in these assays correlate with what's seen in the clinic. So that's what's really important, that the data generated in the assays is representative and can be trusted to support the understanding of the immunogenicity risk associated with the product. And then just to finish, really a summary of the earlier aspects, so the tools which are really supporting the preclinical assessment activities. I hope I've shown you that some of the in silico and in vitro tools that can be used to provide, you see there, a rapid and cost-effective method that really do support the lead selection and characterization. Many of these tools, they take a week or so with the in silico tools, maybe to 2 or 3 months with the early stage expression and some of the in vitro assays. But a lot of this work can be performed in parallel. You don't have to, they don't really have to be an impact on the timeline of your program. It's important, obviously, that work with the most suitable expression host from an early time point. And really, what I've not yet mentioned, but I will mention now, is that the earlier you work with the cell line or the expression system that is your choice of expression system for cell line development, the better. We are always going to see slight differences in the post-translational modifications. For example, if we move between different types of CHO, or obviously from a 293 to a CHO cell. So really important that we do, even with the early research work, work with the product in the same cell line as you would intend to use for cell line development and manufacturing. So a lot of these tools allow us to identify the issues early. If we do identify an issue which really is quite a problem for your product, then we can support with re-engineering. All is not lost if you do identify an issue, but obviously the earlier you do identify the issue, the better. Just a reminder there of what I mentioned about the immunogenicity risk. and are now being a requirement of the regulatory filings to show that you put a strategy in place to look at immunogenicity risk. Know that the more tools we use to support the late stage discovery and early development, the stronger position you're going to be in terms of having that reassurance that you really are selecting the right lead to go on and be developed, go into manufacturing, and ultimately go into patients. So with that, I will now hand over to Alison, who will continue.
So thanks, Yvette. So you've just heard Yvette discuss the use of a developability assessment and how the work her group does can help you reduce risks and increase the likelihood of identifying successful candidate molecules. Now, once a final variant of your molecule or a small number of variants has been selected, it's ready to progress to the creation of a manufacturing-ready clonal cell line and the development of a cGMP-suitable production process. Now, it's really important that the right approach and best tools are selected here too, as biotherapeutics can also fail due to issues in the cell line development and process development stages. So following on from Yvette, I will briefly introduce Lonza's GS gene expression system, a robust, fully integrated, scalable expression system with a proven track record of successfully taking programs from gene through cell line construction to commercial product. And I'm then going to consider next generation biologics, a pipeline of more complex proteins that we are seeing coming through, and introduce GS Piggyback, a transposon-based technology that's part of the GS toolbox and which may be especially beneficial in helping you be successful with such molecules. So let's start with Lonza's GS gene expression system. So Lonza's GS system, put simply, it is a platform for producing recombinant proteins using mammalian cells. And why choose Lonza's GS gene expression system? Well, we have a market-leading expression technology that is combined with extensive Lonza know-how, and the platform is portable and versatile. We use our GS system in-house when we are creating cell lines and developing processes, and it is exactly the same system we license out for others to work with in their own labs. And that means there is extensive knowledge of the system with Lonza scientists, which is accessible by projects being performed within Lonza, and also by those working with the system in their own labs, because we want them to get the best out of the system too. It also means that a customer can do as much or as little of a program as they like, and easily transfer at any stage for us to do work if wanted. It's a robust and reliable technology for expressing a diverse range of biologics. The basic system has been in use for almost 30 years, and its various iterations and additions have been used by us and by many biopharma and biotech companies of all sizes. And the net result is that there are now many marketed products that use Lonza's GS system, and this is in the dozens, and a large number running into hundreds that are in active clinical trials that use the system. Use of Lonza's system can de-risk partnering of programs and increase the value of drug candidates because large pharma biotech know and trust the GS system. So CMC section discussions can end up being reduced. And finally, we continue to improve the system, aiming to meet the needs of the industry as things move forward. And in a few slides' time, I'll try to show you this with a flow diagram depicting when various tools have been added to the GS toolbox. When we say the GS gene expression system, people expect basic vectors and a host cell line, but there are actually a number of components that comprise our GS-Exceed expression system, and this slide takes a look at that. There is the CHOK1SV GS knockout host cell line we routinely use, and there is the Patelligent CHOK1SV host cell line for those looking for enhanced effector function. We've got documented history on the host cell lines and fully tested cGMP cell banks. Then there are various vectors that can be used with the system. PXE 17.5 and PXE 18.5, which we know as our base vectors. They're used for the expression of one or two genes, so they're traditionally used for the expression of antibodies. Then there are constant region vectors, which are based on the universal base vectors, but as the name suggests, they already encode antibody constant regions. There are multi-gene vectors for the expression of 3 or 4 genes, and therefore really useful when expressing more complex proteins with multiple genes such as bispecifics, or when expressing proteins that require helper genes. And there are site-specific conjugation vectors which are used for antibody-drug conjugation. Also, the media and feed platforms we've developed and used form an integral part of the system, and an updated version was actually released just last year. And then there's GS Piggyback, a transposon-based technology that I will also talk more about today. And as well as the physical materials, we also provide access to know-how for those that work with the system in their own labs. Now, this is predominantly either in the form of protocols, which guide you on how to work with the system, or via direct scientist-to-scientist support, so email or phone or even face-to-face if possible, connecting you with subject matter experts such as myself from Lonza. This is because we want to enable those that have in-licensed the materials to get the best out of them. And I should also add that as well as technical support, we do also provide regulatory support at the stages when needed. As promised, on this slide I have a flow diagram that shows the continuous advancements that have been made to the GS expression system since it was first introduced in 1992. For example, the CHOK1SV host cell line was first introduced in 2003, the Exceed host cell line was launched in 2012, and platform upstream media and feed packages have been released for use by GS customers at various stages. Towards the end here, you see our multi-gene vectors, which were introduced at the end of 2018, GS Piggyback, which was added in 2019, and as I said on the previous slide, our latest media and feed platform, GSV9, which was launched last year. Next, I wanted to note that many people associate the GS system with IgG antibodies, but it is much more than just that. The system is used to produce IgA, E and M antibodies, as well as IgGs. And it's also used for a whole multitude of other proteins, including fusion proteins, enzymes, interleukins, hormones, and so on. So we certainly have a lot of experience with a diverse range of molecule types, and this slide looks at that experience. In the left sunburst, our experience with different molecule types in the discovery early development stages are shown. Now I should know that Yvette shared a variant of this sunburst in an earlier slide. This one breaks down the different products into more detail, but you can still clearly see it's not far off a 50-50 split between antibodies and non-antibodies. Now the right sunburst then looks at the data from development, and what you can see here is that antibodies are still currently dominating in projects that have moved into development. But there's a definite shift being observed in the pipeline, with more next-generation biologics coming through. So what if you do come to us with a more challenging protein or project? Well, we do have a number of physical technologies available to help with a number of different challenges whose use can be considered. We have the multi-gene vectors and GS Piggyback. Both can be especially beneficial if you're trying to express more complex, difficult to express proteins, and I'll introduce the latter a little more in the following slides. We also have site-specific conjugation vectors for those interested in antibody-drug conjugation, NS0 cell lines for those wanting to express certain biosimilars, and our intelligent CHOK1SV host cell line for those looking for enhanced effector function. So talking about challenges, I do now want to move on and look a little more closely at one such challenge, and that's expression of more complex proteins. And one of the technologies we have that can help with this challenge, GS Piggyback. But let's start with a few more thoughts about next generation biologics before I talk about GS Piggyback. Antibodies are still predominating in the clinic at the moment, but there is a definite shift in the drug development pipeline towards more complex next-generation molecules. Now, these next-generation biologics usually combine domains from various molecules with engineered elements, so the structure is not something seen in nature. And often these next-generation biologics do not express well in current expression platforms. So in Lonza, we considered the potential need for a different platform. But the real question is for what? Because the array of new formats for the next generation biologics sector is bewildering. And we've even used a few descriptions and analogies to try to describe this need to deal with molecules of so many different sizes and configurations, such as it's like a zoo of molecules. But regardless of the descriptor used, the net result is the same. We can't follow the approach taken in the past, where as an industry concentrated on addressing the needs of a specific molecule type. There are too many of them. Instead, we need to address general needs arising from engineered proteins. So I think you can probably tell from the previous slide, we identified next-generation biologics as an important area of expansion in the industry. And we've been working on a number of projects internally and with external partners targeting the production of these increasingly complex, difficult-to-express molecules. And I should note here that next-generation biologics aren't just a challenge in the upstream. They also create challenges for downstream, especially if they lack an FC region, and they create challenges for analytics, for which many methods have been developed around antibodies. So although I'm concentrating on the upstream area today, we are working in these other areas too. Additionally, as well as physical technologies, there is plenty we can consider around how we design cell line and process development programs of works. But today I want to share with you one new physical technology that's now available in Lonza's GS Toolbox and has applications both for standard antibodies and the wave of next generation biologics coming through. And this technology is GS Piggyback. Transposable elements are DNA sequences that move from one location on the genome to another and are therefore also known as jumping genes. They were first identified more than 50 years ago by a geneticist called Barbara McClintock when she was working with maize plants. And just to clarify a couple of terms used when talking about transposable elements. A transposon is a mobile DNA element, and the transposase is the enzyme that mediates this mobility. Now, typically in nature, the transposon and the transposase are the same. So that is the transposon encodes the transposase, which then moves that piece of DNA around. If we focus in on the use of transposon-based technologies in our industry, and in particular on our GS Piggyback system, the piggyback transposable element was first derived from the cabbage looper moth, and the system was subsequently reconstituted for use in other cell types by separating the transposon from the transposase. So how does it work? Well, put simply, you flank the genes for your product with inverted terminal repeat sequences, or ITRs, and this is now your transposon. You then co-transfect this transposon plasmid along with a transposase, which is normally supplied as a messenger RNA. The transposase recognises these ITRs, binds, and cuts out that piece of DNA, and it then facilitates the pseudo-random integration of that piece of DNA into another chromosomal site. And I use the term pseudo-random because they preferentially target sites with particular sequences that are associated with stable, highly expressed genes. The messenger RNA will have a short half-life, so once the transposase activity has finished, the recombinant genes should be stably incorporated into the genome. So the system is working via a cut and paste mechanism. The DNA cargo flanked by the ITRs can be very large, and as a result of its targeting particular sequences, there is an expectation that this will ensure stable, high expression. And because of the way it leads to stable, high expression, it is of special interest to more complex proteins, which can often be more difficult to express. Also, there is potential for the technology to improve cell line stability. So I want to move on to look at two case studies undertaken at Lonza. Let's start with the first case study. Now, the aim of this work was to demonstrate how the hyperactive super piggyback variant can be combined with Lonza's GS gene expression system to create stable CHO pools with improved expression of a challenging monoclonal antibody. Now, the initial step in this work was to select a model antibody that was difficult to express, then to construct vectors containing the ITR recognition sites for it. Stable CHO pools were then constructed using these vectors on multiple occasions, and other variables were assessed, including things like the amount of transposase. Finally, pools from each experiment were assessed in a scaled-down shake flask model. So let's start with step 1, selection of that difficult-to-express model antibody. So we'd constructed a panel of engineered antibodies and expressed them using the GS system in stable pools, and these were then assessed in 10-litre bioreactors. And in this graph on the left, you can see the product concentration results from those bioreactors. If you look on the far left of the graph, you can see a molecule called AMS002, which was the lowest expressing antibody in this panel. And it was this molecule we therefore decided to use for this case study. So we created a GS piggyback double gene vector for expression of that AMS002 molecule, and then on 6 separate occasions constructed 3 stable CHO pools using this vector in our CHO-K1SV GS knockout host cell line, which were then assessed in a scaled-down shake flask model. At all times, controls were also included, as you will see in the results. So let's look at some of the results, and we'll start with the graph on the left. Now the grey bar on the right of this graph shows the original performance of this molecule in the 10-litre bioreactors. You can then see that we saw similar performance in the shake flask for this molecule when the same random integration approach was taken, and this is seen in the blue bar labelled as the random integration control. For interest in turquoise, so that's the bar labelled as no messenger RNA control, we've also included data from an alternative control, where we used the GS piggyback vector but did not add the transposase. And then if you look at the first bar, so the maroon bar on the far left of the graph, you can see that on all occasions, use of piggyback resulted in a substantial increase in titer in the pools, with an average increase of 65% compared to the random integration controls. If you now look at the right-hand side graph, we did have a look at varying the amount of transposase we added. We looked at 25%, 50% and 100%, as well as our standard 10%. And you can see that increasing it above the standard 10% we were using had negligible benefit. So we continue to use 10%. And this graph looks at an interesting observation. When pools are created using GS piggyback, so that's the maroon bar on the far left, they recover faster than both the random integration controls, so that's the blue bar in the middle, and when we transfected using the piggyback vector but didn't add the transposase, so that's the turquoise bar on the right. On average, what we've been observing is that GS piggyback pools recover around about 4 to 6 days faster. So now let's take a look at the second case study undertaken at Lonza. And again, the aim of this work was to demonstrate how the hyperactive super piggyback variant can be combined with Lonza's GS gene expression system to create stable CHO pools with improved expression, but this time for a diverse range of molecules. So vectors containing the ITR recognition sites were created for a selected range of proteins, and stable CHO pools were then constructed using these vectors. The pools for each molecule were then assessed in a scaled-down shake flask model. Like in the first study, we created GS piggyback double gene vectors for expression of the various molecules, and we then constructed 3 stable CHO pools using each vector in our CHO-K1SV GS knockout host cell line, which were then assessed in a scaled-down shake flask model. As previous, controls were also included. Now when you look at this graph, each colour represents a different molecule. Then the first bar for each molecule are the results for when GS piggyback was used. The second is for the random integration control. And the third from when we transfected using the piggyback vector but didn't add the transposase control. And the different molecules we assessed were that difficult to express AMS002 antibody, a standard antibody, a complex antibody which had a common light chain and 2 heavy chains, which differed in that one of them had an adjunct. And finally we assessed a 4-chain bispecific. Now, these are preliminary data, as we've yet to repeat the experiment multiple times in the way we have for the AMS002 molecule, but we did want to share the data. As normal, we do see an increase in titer when piggyback is used for the AMS002 molecule. And for the other 3 molecules, a significant increase in titer was observed for 2 of them, and a slight increase in titer was observed for a third. And finally, in this section on piggyback, I want to focus in on the complex antibody and look at assembly as well as titer, and also take a quick look at the impact gene order can have when using the multi-gene vectors. So let's start with assembly. For this complex antibody molecule, we performed LC-MS analysis as well as measuring product concentration via octet. Now, the data points above the product concentration bars are therefore for percentage heterodimer. And what we can see from this is that the increase in product concentration with GS piggyback compared to both the random integration and no transposase controls does represent assembled heterodimer rather than just an increase in fragments. Also, for the complex antibody and the bispecific, we had to construct multi-gene piggyback vectors. So as it is possible with the multi-gene vectors, we had a look at changing the gene order for one of the molecules. And you can see here that changing the gene order can have a substantial impact on product concentration achieved. And I will add that use of GS piggyback resulted in an increase in product concentration over the random integration control for both gene orders tested. So all in all, we're seeing some great results with piggyback. And now I'd just like to finish off today with some concluding thoughts. At Lonza, we offer a service and product portfolio to ultimately enable our customers to help their patients. Today, we've discussed the use of a developability assessment to address potential issues with your molecule early, thereby reducing risk and maximising the chance of identifying a candidate molecule that will go on to be a successful biotherapeutic. And following this, we've touched on parts of our mammalian development capabilities, which can be used to further help you be successful with your molecule. In particular, we focused on one aspect of our product portfolio for mammalian development, and that's the GS gene expression system, a robust, fully integrated, scalable system with a proven track record of successfully taking programs from gene through cell line construction to commercial product. And finally, as part of the GS system family, we've introduced GS Piggyback, a technology that I've hopefully shown can substantially increase the expression of a challenging antibody, a complex antibody, and a bispecific from stable CHOK1SV GS knockout pools. So that just leaves me to say thank you for listening, and Yvette and I will happily take any questions you have.
Yvette, Allison, great presentation with a ton of information and data, so thanks for sharing that. I know the audience probably has a bunch of questions that they're ready to ask after all that. So with that, just for the audience so they know, the Q&A is coming up in just a few moments. And I want to remind you again to send in your questions for Allison and Yvette. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. All right, without any further ado, let's get to the Q&A session and get to those questions. Bear with us for just a moment as we transition into the Q&A. All right, everyone, thanks for joining us for the Q&A. We got a bunch of your questions already, so let's get started. The first question is going to be for Yvette. Yvette, one of our audience members would like to know, optimally, how many molecules of monoclonal antibodies would you take for the developability assessment?
Okay, so thank you for that question. It's a really good question because I think the honest answer is there's no set number. It really depends on everyone's capabilities and the number of molecules that you have at a particular time. Obviously, the in silico tools do lend themselves to higher throughput. We may routinely screen, say, between 2 and 10 candidates, but even if you have just a single candidate, the in silico tools still add value. we can screen more, but I would say a typical number is around, say, 2 to 10 candidates for in silico stages, and then we may reduce that down to, say, 5 products or so for perhaps some of the expression or some of the in vitro immunogenicity. But the honest answer is there is no real set pattern. Everyone has different requirements, and a lot of it will come down to kind of time and actually just how many molecules you have in the first place.
All right, thank you for that. Allison, next question is for you. An audience member would like to know, should I test adding different amounts of transposase if using the piggyback system?
Okay, so you can, of course, look at this if you wish, but based on published studies plus our in-house work, we concluded that 10% weight per weight is the best transposase load in our system when generating high-performing pools, with additional transposase actually offering very little benefit.
All right, thank you, Alison. Yvette, back to you. Is EpiBase publicly available?
Yeah, so I assume this is referring to the in silico tool. It's not publicly available. It is a Lonza proprietary tool. So we offer it for either in 2 different ways, either a straight fee service with our clients bring the sequence to us and we perform the analysis, or it can also be available as a server and our clients can take the server in-house and perform the analysis themselves. But no, it's currently not publicly available.
All right, thank you for that. Allison, an audience member, has a question that says, in most cases, it looked like the titer for the controls where you did not add the mRNA were lower than those with random integration. Why do you think that is?
That's a really good question. So we've discussed this internally, and 2 things that have been suggested as possibly having an impact are, well, firstly, that the piggyback vector used for the no transposase control is circular, whereas the base vector used for the random integration vector is linear. Second thing that we've considered is that the piggyback vector will be bigger than the base vector due to the presence of the ITRs. And as we add the same weighted DNA regardless of the vector, this will have resulted in there being less copies of the piggyback vector compared to base vector in the same size sample.
Okay, thank you. Yvette, are there likely any immunogenicity against hybrid peptides that contain, for example, 44 amino acids?
I guess the answer to that question is that there could be. So I think so long as the size of the protein is over a certain length, we use certainly in our in silico tool 10 mer, I think typically up to about 13 or 14 mers. Then any protein, really any that's of a suitable size, could contain epitopes and therefore could be immunogenic. Certainly hybrid proteins, so I'm kind of taking that to mean obviously either a fusion or a combination of a couple of different proteins. Potentially the join or where the joining site and such of 2 proteins could well be immunogenic. So the bottom line is that it could be. It could certainly be immunogenic. Really, any peptide over about 12, 13 amino acids could actually be immunogenic. So it certainly warrants some assessment.
All right, thank you for that. Allison, back to you. This is a good question. What expression levels do you typically see with Lonza's GS system?
Okay. In order to answer that, I should actually start by reminding everyone that we do have a platform upstream process, and that includes platform medium feeds that have been developed specifically to get the best out of cell lines made using Lonza's GS system. So when we make a cell line, and at the end of cell line selection, we then evaluate that lead cell line in 10-liter bioreactors. We do so in our platform process. So this is dropping straight into the platform. There's no process development work. So anyway, when we do this, in those 10-liter bioreactors, we typically see between 2 and 6 grams per litre for an antibody with an average of 4 grams per litre. When we have then done further process optimization work, we've seen up to 10 grams per litre. For non-antibodies, we also typically see in the range of 2 to 6 grams per litre, but the average is slightly lower at 3.3 grams per litre. And I guess it might also be worth commenting on pools, because these can be helpful to feed other development stages. With these, on average, we've seen around 2.4 grams per litre for antibodies, but we have seen over 6.5 grams per litre. And I guess one final comment I feel I should make is that over the years, we have observed that the biggest influence on product concentration appears to come from the amino acid sequence of the antibody. And so for antibodies, the more engineered the antibody is, the more likely it is to be challenging.
All right, thank you, Alison.
Yeah, and
Yvette?
Just jump in.
Oh, absolutely.
Sorry,
sure.
I was actually just going to jump in and add to Alison's comment, if I may. I think Alison sums it up brilliantly. A lot of it is due to the sequence. A lot of people, we see aggregation as, I think, a common example, obviously, and can quite often influence the titer. I think knowing how some areas and departments work, quite often the aggregation problem can just be pushed down the line. It can be pushed from the cell culture people to the purification to the formulation group, to try and stabilize the molecule. But all they're doing there is essentially putting good or potentially good product down the drain, that's aggregated. So I totally agree with Alison's point there of what really contributes most is the amino acid sequence.
Alrighty, thank you. And Yvette, stick with me. We have a question for you, one that I think is probably of pretty great interest to the Gen audience. An audience member asked, is the data generated by your immunogenicity assessments suitable to include in regulatory findings?
There is a very quick and easy answer to that, and it's yes. So the likes of the FDA and other regulatory authorities are familiar with the sorts of assays that we're performing, both the in silico and the in vitro, and it is absolutely taken into account during filings. We've actually had a couple of recent examples where the clients have been ready to submit their INDs with the FDA, and they've actually been asked to go and get some of these in silico and in vitro data to add and include as part of their submission. So yeah, the regulatory authorities are very familiar with these sorts of activities.
All right, great. Alison, back to you. Are there any major changes to the transfection or cell selection protocols when using GS Piggyback?
Yeah, this is also a very quick one to answer. So there are some differences in vector construction, and of course there's a need to include the transposase messenger RNA in that transfection step. But once you pass this, the workflow for cell line construction is as normal for a cell line development workflow.
All right, thank you. Yvette, back to you. Is in silico immunogenicity assessment sufficient to move leads forward? And when would you suggest running in vitro immunogenicity assessment?
Okay, so let's start with the in silico. So the in silico tools are quite often performed when there are a large number of candidates. So they can be a very efficient way, particularly when combined with some of the other early risking tools or de-risking tools which are out there. So it can be a very efficient way to, at an early time point, stop working with the higher risk candidates and focus on the lower risk candidates. So I think, yes, the answer is yes. Potentially, you can make selections of your lead candidates based solely on the in silico immunogenicity. Obviously, it depends perhaps on how different the scores are. They need to be statistically significant from each other to really have to increase the confidence in essentially stopping work with a candidate. And my mind's gone blank. What was the second part of the question, please?
It's when would you suggest running the in vitro immunogenicity?
Yeah, when would you use the in vitro? The in vitro really can be used at any time point, and we have such a large number of different assays which really come into play at different times. So a lot of the assays are routinely used to support variant screening and to support lead selection. So they can be used using either some early stage material. We don't need much material for it. Some of the other assays, particularly those looking at impurities, may be used later during cell line development or manufacturing, obviously to look more closely at the product. Perhaps if there are any biosimilar developers as well, the assays can kind of be used during cell line development activities, because it's important even with biosimilars that you're showing that the innovator product has a comparable level of immunogenicity risk to the biosimilar product. So really the different assays come into play at different stages of development.
All right, great, thank you. Alison, back to you. Can the piggyback system help to address challenges of cell line instability from the product characteristics point of view?
Yeah, it's an interesting question, and it's something that I hope that we will be able to answer very shortly. Piggyback's still relatively new to us at Lonza, but Piggyback has been around for a while, so there's already quite a lot of reports about it in the literature. And when you look in the literature, there are a few reports around stability, but of course most of them concentrate just around talking about productivity and maybe a little bit about growth. Because of course that is the theory with the piggyback system, where it's integrating into more stable regions of the genome. You're expecting to see more stability when it comes to productivity. But we are ourselves conducting studies at the moment, so we would hope to very shortly be able to answer that question on whether it could also have any impact on on product characteristics from a positive viewpoint.
All righty, thank you, Alison. Yvette, do I need to perform all the risk assessments you mentioned?
I think in an ideal world, yes, perhaps at different stages, but we appreciate that everyone has a different strategy and perhaps different levels of funding. I think the really important thing is that people are looking to see, are aware of the de-risking strategies and putting a plan in place to include as many as they can. But they really all come into play at different times. So I would say in an ideal world, yes, start with a couple of the in silico tools, the immunogenicity, the manufacturability. Both are very quick tools and get you some useful information for very minimal investment of both time and cost. Then perhaps move to the expression, but again, it depends on your capabilities. Obviously, some people may have their own expression, but certainly include the in vitro immunogenicity. So there's really no set workflow of performing these. We would certainly encourage everyone to try and include as many of these tools as possible. But it obviously is, everyone works differently and has a different strategy. But I would strongly encourage you do as many of them as possible at some point during the late discovery and early development. And really, the earlier you can do these, the better. I mean, we've had cases where our clients have been in phase 1 and they've seen an immunogenicity issue. That is obviously far too late for many people. Use particularly the in vitro immunogenicity tools to support your development, to support your variant selection, and really the earlier the better. We very much appreciate the speed of getting to clinic and to the market is obviously vitally important. But some of these services take just a couple of months. They can be run in parallel with other activities. Run some in vitro immunogenicity in parallel with some efficacy studies, and it really does add value to your program, and should stop you getting to the stage or being in the position where your product may have an unwanted immunogenic response, and potentially you have to go back a long time in your development and redo a lot of work.
All righty, thank you for that. Alison, Dan would like to know, why was the scale defined at 10 liters for the upstream workflow?
Okay. So when we do cell line selection, the final vessel type that we normally use in that process is going to be an amber 15. And then we will normally look at least the lead cell line in lab scale bioreactors and look at it in the platform process in those lab scale bioreactors. Now, our lab scale bioreactors are 10 litres. and that is partially historical. So we have 10-litre glass bioreactors that were originally designed to be scaled-down mimics of our very large stainless steel vessels. These days, of course, we're using more single-use bioreactors more and more, but what we've found is those 10-litre glass bioreactors can, if you operate them in a particular way, can also be representative of the single-use bioreactors and the way in which we work with them. So put simply, we've got 10-litre vessels that are very good models for the various manufacturing vessels that we use within our network. So it gives us a good lab run through of the production process to see how a cell line will perform.
Alrighty, thank you. Yvette, we have another question for you. When should the different assessments be performed?
Well, ideally, the in silico services should always be performed first because quite often they're more suitable for the higher throughput. But really use these before, or as much as possible, use these services before making your final lead candidate selection. Use the in silico can often be used to support late stage discovery because they're high throughput. We can screen a higher number of sequences, but ultimately, please use these assessments before you are making your final lead or final choice in your lead. Make sure you've de-risked your candidate as much as possible. But having said that, obviously some of the immunogenicity in vitro assays can be used later. As I think I've just alluded to, some of them are more suitable for use during development and manufacturing as well.
All right, and it looks like we have time for one last question for Yvette. An audience member would like to know if they can use transient or stable pool material in the in vitro immunogenicity assessments, or does it need to be clonal?
Yeah, absolutely. You can use transient or stable pool. As I've just mentioned, a lot of these tools, they should be used to support lead selection, and obviously, a lot of people are not going to go into clonal cell lines until you're ready to go with your selected lead. I think all I would say is if you're using the screens to essentially to help with your candidate selection, you're screening different sequence or product variants, do try to use material which is being expressed in the same way, whether it's transient or stable pools. It doesn't matter if it's been expressed in 293s or CHOs or even E. coli. Just try and have it all made or expressed and purified in a similar manner so that you've got a good degree of comparability between the different variants that you're screening. So certainly comparability in terms of how they've been expressed and purified.
All righty, thank you. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues who you think might be interested. I'd like to thank Yvette and Allison again for their very informative presentations, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Lonza for sponsoring this webinar. Hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy.